let's get started. So, how you guys feeling about this? Better on this one than last one or not? More material, less material? What do you think? More. Okay. More what? Okay. All right. I try to divide them up so that there's at least what I think is reasonable amounts of material, but every class is different sometimes what they think too, so that's interesting. It may have to do with how fast I talk too, so I don't know. Okay, so what would you like to know? Yeah? Uh-huh.
Things as an educational way to present the mechanisms.
Yes?
Would you also like us to know the exact differences between the two things?
Okay, so her question, I'm going to repeat your question just so people can hear. So the question was, in class I talked a lot about various mechanisms that were prokaryotic or eukaryotic, and the question is, did I want you to know the details of each of those mechanisms? I think that's the question. Is that?
Just the differences.
Okay, just the differences.
Like if you are going to say, is this a prokaryotic mechanism or just a eukaryotic?
Okay, so her question is, am I going to say, is this a prokaryotic or a eukaryotic mechanism? In general, I'll be honest with you, I don't usually ask a question, is this prokaryotic or eukaryotic? But I would not uncommonly ask something that might be about prokaryotic or eukaryotic, and I think that you should know something about that. So for example, I mean, I just, off the top of my head, if I said, okay, tell me, how would you describe what telomerase does, and why is that relevant? Okay, you would say, well, telomerase lengthens the end of eukaryotic chromosomes, right? So that would be part of your answer in doing that. Does that make sense? So, but I'm not going to go in any more depth, certainly, than what I've talked about in class. And in fact, I think you'll find in general that when it comes to prokaryotic, eukaryotic, I'm probably not nearly as detailed as I actually am in class. Yeah. Yes? I'm sorry? I can't hear you. Yes, come to it. Okay, so a good question. So I didn't really talk much about that, so let me just answer her question. Her question said, I talked about the catalytic triad of the aspartic acid and histidine and serine, and I didn't really say much about aspartic acid. So how does that come up? So where am I at? A little higher? Right there. Okay. And I had a nice figure there. Okay, I like this figure a lot. Okay. This is showing up close and personal what's happening in the active site of the serine proteases. And aspartate is right there. Aspartate is, and what I'm going to tell you here you're not responsible for, but I'm answering your question. Okay. So aspartate is negatively charged. And that negative charge means it's obviously electron rich. And here's the histidine. This histidine ring has electrons flowing around in it. Okay? When the proper substrate binds in the S1 pocket, the enzyme changes shape very slightly. And that slight shape change causes these three things, aspartate, histidine, and serine, to become closer together. Well, if you put a negative charge right next to this ring full of electrons, what's going to happen? The electrons are going to avoid the charge, and they're going to move to the other side. The movement of the electrons to the other side makes the ring more negative right here, and that ring really loves that positively charged proton that's on serine and grabs it. So the role of aspartate doesn't seem to be very important. It actually is. But its role is to push those electrons over there so that this will pull the proton off. Does that make sense? It's kind of a cool process. Yes? Okay. So her question had to do with the sodium-potassium pump that I talked about. And her question was, what purpose does it serve? Because the cell's wasting a lot of energy pumping sodium out and potassium in. Why is it bothering to do that? Okay? So the answer to that question is that the cell has got to maintain osmotic balance. So like the dialysis tubing experiment that I talked about in class, if you take dialysis tubing and you fill it full of protein and you put it into a solution of water, the dialysis tubing will swell until it bursts because the protein can't get out, but the water can get in. And so the water diffuses in to try to dilute the protein out, but the protein can't go out, and consequently the dialysis tubing will burst. Well, cells are an awful lot like that dialysis tubing in the sense that they're full of proteins and they're full of ions and various other things, and those can't make it out of the cell either. So a cell has to play some tricks with the balance of ions in order to keep itself from bursting. So that's what it's doing, is it's actually messing with the ionic balance so as to stop the diffusion process, because remember, water can move into cells very readily across the membrane. And so this is done to keep water from coming into the cells. Yes? Yes. Okay, so the anticodon that's there. So let me say a word about the anticodon. All right, that would be down here. And was it? Oh, back here. Okay. All right. Forgot where I had it. Okay, and we had tRNA structure. Okay, so I'll start with this. Somebody asked me earlier today, do we have to know that structure? No, you don't need to know the structure. Okay, it's what I describe as a clover leaf, and you can think about it as either 3 or 4 leaf clover, whichever way you want to is fine. Okay, there are 2 structures on here that you should at least know what they do. You don't have to draw them or anything, but you should know what they do. One is up here. The 3' end is where the amino acid gets attached. And the other is at the opposite end of the molecule, down here labeled 2, and that's where the anticodon is. Now, to understand what an anticodon is, we have to first of all understand what a codon is. So a codon is a 3-base sequence that's in the messenger RNA. So the AUG that codes for methionine is a codon. All right? The anticodon is a 3-base sequence that's down here that's complementary to a sequence in the of the codon. So something that's carrying methionine up here is going to have a sequence that's complementary to the AUG down here. Does that make sense? So that's what an anticodon actually does. Nothing more than that. Okay? But when I talked earlier today about the fact that those base pairs had to be right, or EFTU would take the tRNA away, this is the half of the base pairs that it's concerned with, the anticodon pairing with the codon. Okay, yes? Okay. Okay. So I pointed out that I thought you should be able to recognize the structure of cholesterol, and I also showed the bases like guanine and cytosine and things like that. And her question is that something that I should expect that you should be able to draw? What do you guys think? You want to draw them? No. So you don't need to draw them. And I'm not going to hold up a cholesterol and a guanine and say which one is which. I'm not going to do that. So I'm talking about very, very general things in terms of structure. Yeah, I don't do much in terms of structure in this class. So when I'm talking about things, I'm talking about it very much in general. So, I think you should know, for example, that a purine has 2 loops and a pyrimidine has 1. Because the 2 go with 1, and then it's always 2 and 1 and 2 and 1, so they always stack up right inside of a double helix. So you always have a purine with a pyrimidine. A goes with T, G goes with C, etc. Those sorts of things. That's, very basic kinds of stuff like that. Yes, question? Oh, yeah, that's how the factor-independent hairpin stops the synthesis of RNA in transcription. So that's a good question. Excuse me. So factor-independent occurs here. And this is always one that's a little hard for students to get their heads around a little bit. Okay? So to understand this one, we have to understand what's happening in the cell. All right? So during transcription, what's happening? Well, I have an RNA polymerase, and I'm going to go back to the transcription bubble here. Let's see. Here's the transcription bubble. Okay? And we call it a bubble because that's what it looks like. The RNA polymerase is right here on the very front, and it's moving towards the right. Okay? And it's actually, in this case, it's copying the top strand is what it's copying. All right? Doesn't really matter for our purposes, but it's moving along copying the top strand. Okay? The RNA is hanging off the end, and as we get further along, we're going to see a long tail of many bases that are hanging off of here. Okay? Well, that's what's actually being shown right here. All right? So here's the DNA that's being copied, and here's the RNA tail that would be hanging off of it if this DNA extended all the way over here, which it actually does. Okay? The RNA tail is hanging off of it. All right? The RNA polymerase is sitting right here. It has just finished synthesizing. It has made all of this. It's made this. And now this thing all of a sudden goes, whoa, I see a complement. Doing, 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 doing. And it just automatically forms. Base pairs really like to form. They will form all by themselves if there's a complementary region for them to form. And that's what's happened here. So this sequence in DNA got copied. This was the sequence that was in DNA. It's now in RNA. But the difference here is once this is now abundant like this, or out here like this thing starts pairing. Well, when it pairs, it forms something that's like a jack. It's just like a jack on your car. And so it actually lifts. The polymerase was sitting here, but now it's lifted the butt end of the polymerase up. And the polymerase was sitting on the DNA, but now it no longer sits on the DNA. So you've taken the polymerase off of the DNA, and when you do that, everything falls apart. And it's a little hard to conceptualize, but the important thing is that the automatic formation of this structure, it causes a physical lift of the RNA polymerase off of the DNA. Okay? When we look at what's happening in the factor dependent, here we have the row that's catching up with the RNA polymerase. What row does when it catches up with the RNA polymerase is exactly the same thing. It simply physically lifts it up off of the DNA and everything falls apart. So in this case, a protein is causing that to happen. In the other case, it was the RNA itself that caused that to happen. Okay? Okay. Yes? Say it again now. Okay, so the stop codon, remember, occurs in translation, not in transcription, right? So what's happening here is there's really a race that's going on. So this guy does move, Rho moves faster than the RNA polymerase does on average. All right? So the RNA polymerase gets a head start before Rho actually gets on there and starts racing. But Rho catches up with it because the RNA polymerase doesn't move quite as fast, and it tends to slow down when it hits GC regions. So if there's a bunch of GCs right here, it'll slow down. That gives Rho a chance to catch up. Now, your question is, how does it know where to stop it? It just stops it where it catches up with it. So with Rho, there are not specific stop places. We'll see general places where they will stop, and they'll usually be close to GC regions where the Rho catches up with it. Does that help? Hope I answered your question. Yes, back to back. Okay. So P53 in eukaryotic cells. P53, as I said, is one of the most interesting proteins in all of biology, I think. Okay? Because P53 is a quality control for DNA replication. Quality control. And it literally does just that. So remember, we have the cell cycle in a eukaryotic cell. That is, we go from G1 to S, G2 to M, okay? And the synthesis of DNA occurs during S phase, and it's essential that the synthesis of DNA progress properly. That is, that if there were problems that arose during the synthesis of DNA, that they get fixed. If they don't get fixed, then the cell has a defective DNA, basically. That defective DNA can have mutations that can then give rise to cancer and other problems. Okay? So P53 can't ensure that there are no mutations at all. I don't want to leave the impression that it says if there's a mutation, we stop everything, because that's not how it works. P53 is there when it recognizes that there's big processes in replication that have gotten stuck. So, for example, there are still replication forks in the cell at the place where replication should be done. That would be a signal to P53 to say, whoa, something's not done here, let's stop everything. So it stops the cell from progressing to the next phase of the cell cycle, and therefore keeps the cell from progressing onto dividing. All right? So P53 stops at that point. So if it sees that there's something that hasn't finished or something is stuck or whatever, it stops. And what P53 does at that point is it induces the synthesis of repair proteins, DNA repair proteins. And there's a ton of them that get made at that point. And the repair proteins have the job to go out to those places where replication has stopped to see, is there anything here I can fix? Are there DNA adducts, these big honking things that get stuck onto DNA? Can I take some of those off? Are there regions I can repair? Are there things that I can do to allow this replication to proceed? And if the repair proteins can do that, then they do that, and replication goes on, and P53 says, okay, go forward and divide. Okay? If the repair proteins cannot fix the problems, then P53 will cause the cell to commit suicide, and that process is called apoptosis, A-P-O-P-T-O-S-I-S. People mispronounce it, call it apoptosis. It's not apoptosis, it's apoptosis. Okay? And that's what P53 does. Pretty amazing protein. Okay? Other questions on that? Is that okay? Okay. Yeah? So in eukaryotic cells, termination is more complicated than it is in prokaryotic cells, partly because in eukaryotic cells, transcription itself is quite a bit different than it is in prokaryotic cells. So I won't comment on termination in eukaryotic cells, but I will say that it is somewhat different, yes. Yes? The Tata box, sure. Okay? So, oh, I was right here. What's that? Okay, so we had this guy right here. This is what you're talking about, I think, right? Okay, so this is actually called the Pribno box in prokaryotes. It's called the Tata box in eukaryotic cells, but you can call it the Tata box if you want. I frequently do myself. But suffice it to say that this box that's here is something that people discovered early on when they were studying, well, how does transcription occur? What controls transcription? And they knew there had to be something that was very specific that told the cell where to start and where to stop transcription, because they would get specific sizes of messenger RNAs that would be made. So they knew they had to have specific starts and stops, and that made sense, but they didn't know how it happened. So they started determining the DNA sequence of the regions near where transcription occurred. So these are all different genes in E. coli whose sequence has been determined, and in every case you can see the start base labeled in yellow. In some cases it's one of possibly 2 or 3. It's usually an A or a G. You see this is an exception with a C right here, but it's usually an A or a G. Okay? Well, when they lined those up and they looked ahead of that sequence, they said, whoa, look at this AT-rich region that's very close to the start site. The person that discovered that was somebody named David Pribnow, and he said, this isn't probably just a coincidence that there's an AT-rich region right next to the start. It probably has a function. Okay? Well, the function turned out to be that it was a recognition for the sigma factor of the RNA polymerase to come and bind, and for RNA polymerase to open up the strands and start replication. That's what's going on here. Okay? So the Pribnow box carries that function. A very similar box called the Tata box occurs in eukaryotic cells, and it's called Tata because it's T-A-T-A-A-A. Okay? Now, as I said, eukaryotic cells are a lot more complicated. The prokaryotic cells often have this minus 35 region that you can see here again. It's kind of hard to see a pattern of sequence. You see this TTG, TTG, TTG appearing throughout there, but it's basically a region that looks something like this overall. Okay? And it's about, and again, you see these are not exact. You can see this is 1, 2, 3, 4, 5, 6, 7, 8, 9 bases away, even though we call it the minus 10 region, right? Or this one is 1, 2, 3, 4, 5, 6, 7, 8 bases away. Okay, but it's approximately 10 bases away. That's where the Pribnow box is. And this is approximately 35 bases away from the start site. So all these are regions that RNA polymerase is recognizing thanks to the sigma factor, binding to, and then starting transcription right next to it. Remember, the transcriptional start site is not here, it's actually over here. Okay? Yes? Okay. The S1 pocket in the serine protease. Yep, sure. Okay, so S1 pocket. There is a lot of stuff we've covered, haven't we? All right. And let's go back to that figure that I just had. Wow. What was it? Is it up one? Okay, here we go. All right, so here's that catalytic triad. Inside, we're looking deep inside the heart of this serine protease chymotrypsin, okay? So we're looking very up close and personal in chymotrypsin. The question is, what's the S1 pocket? All right, so the S1 pocket is just a region of this that's right next to this active site, and it's just like a little cup. It's a region that's part of the enzyme. And this little cup has structures in it that allow it to bind to amino acid side chains. So remember that this enzyme is binding a polypeptide, which is going to have side chains. Okay? And the question is, do any of the side chains fit into that cup? Well, if one of them fits in the cup, the enzyme knows it has found the proper thing for it to cut. So in the case of trypsin, the cup looks for arginine or lysine. And if it sees a side chain of arginine or lysine, the cup goes, oh, I've got it. The protein changes shape very slightly. That's what causes these guys to come together, and catalysis occurs. If it doesn't bind something that the cup would normally hold, which would be lysine or arginine in the case of trypsin, if it doesn't bind to it, then the enzyme doesn't change shape and no reaction is catalyzed. So it's because of the S1 pocket that specificity happens. The S1 pocket determines where the enzyme cuts, because it cuts right next to that thing that it just bound to. That's right. That's right. So the right shape bound into the cup causes the serine protease to undergo a slight shape change, and that slight shape change causes all the things that we talked about earlier to happen. The movement of electrons, the breaking of the peptide bond, and so forth. Yes?
When you were talking about the signal before,
Yes. Okay, so the question is, what is a sigma factor that I talked about in the last slide? So the sigma factor is a protein that binds to the RNA polymerase and helps it recognize promoters. The sigma factor is a protein that binds to RNA polymerase and helps it to recognize promoters. So the Tata box, in the case of the Pribno box, is a promoter. That's correct. I think I might have had one figure. Let's see. Let's see. You got it? Okay, so I have it just there. I guess I don't have a figure of it. Yeah. Okay, it is on there. Okay, yeah. So for the trip operon, well, I explain attenuation, certainly. So attenuation is a process that prokaryotic cells use to control an operon. So what's an operon? Well, remember that an operon is a collection of genes on a single messenger RNA. They're also a collection of genes under the control of the same promoter. That was the definition I think I gave you. Collection of genes under the control of the same promoter. All right. Now, in the case of attenuation, attenuation occurs in operons of prokaryotic cells that relate to amino acid metabolism. That is the synthesis of amino acids. Okay? The cell wants to be making enzymes to make amino acids when it needs them, and doesn't want to be making enzymes to make amino acids when it has plenty of them. So it needs a control mechanism that tells it, do I have plenty or not have plenty, and the process either cause synthesis to occur or stop synthesis from occurring of those enzymes. Okay? Well, the trip operon control is controlled by attenuation, and attenuation senses how much tryptophan is present in the cell. Now, what happens in this process is that when the concentration of tryptophan is high, okay, back up, I'm getting ahead of myself. The trip operon has 10 genes in it. It's big. It's very big. All right? So the cell doesn't want to waste energy on making those proteins unless it needs them. Okay. So, the synthesis of the RNA for the operon always starts. So initiation is not controlled. It's always going on. It's always initiating. The question isn't whether it starts. The question is, where does it stop? Where does the synthesis of the tryptophan operon RNA stop? That's the most important thing. So the trip operon is controlling termination. Attenuation is controlling termination. Not initiation, it's controlling termination. All right. So it turns out that there are two possible structures that the RNA of the tryptophan operon can form. So remember, initiation starts. So this is the RNA for the tryptophan operon. It's already gotten up to about 150 bases, all right? And at this point, either this structure can form or this structure can form. When the concentration of tryptophan is high, the structure on the left forms. When the concentration of tryptophan is low, the structure on the right forms. Now, the structure on the left is a terminator. When the concentration of tryptophan is high, termination stops before it synthesizes the whole operon. When the concentration of tryptophan is low, the structure on the right forms. It doesn't stop. In fact, it encourages the polymerase to keep going and synthesizing more of the operon, and the entire operon is made. So the result of this is that excuse me, the cell's need for these enzymes to make tryptophan are made when needed, that is, the anti-terminator forms, and they're not made when not needed, that is, when the concentration of tryptophan is high and this terminator forms. That was a mouthful. Does that help? Okay. If you approach it by thinking about what the needs of the cell are, you can figure out what's there. You're not going to draw these, don't worry. Yes, sir. Do what? What about them? Do oligosaccharides only come off of integral proteins? The answer is no. Okay? So let me go back and show you a figure. Let me think about that. Where would I talk about that? Membranes. Okay. So this depicts the schematic I showed you of several proteins we find in membranes. Integral. Okay. The embedded, okay, the peripheral, and the associated. And there was one other one that I described, and that was the anchored, okay? So this guy will, in fact, if we're looking at the outside the cell down here, this guy, the integral, will have oligosaccharides on it, okay? You know, as I'm saying this, you are right. And I'm thinking of the embedded. I'm sorry. I'm thinking of the anchored. But the anchored are not attached to oligosaccharides. They're attached to lipids. So the integrals will be the only ones that will have that. Yes. And the reason is that this is the only one that projects all the way through. Yep. And remember that the oligosaccharides are only on the outside part of the cell. None of these guys here have a surface that faces the outside of the cell. Yes? And those are what make up the antigens for blood typing. So the oligosaccharides make up the antigens for blood typing. Yep. Yes? Okay. Well, he said, so the question is, I didn't say much about these other proteins in terms of function, so except for the integral protein. I will say a little bit about some of them later, but I'll just, since you mentioned it, I'll just mention it to you here. So 3 and 4 play some very important roles in signaling. So cells have to communicate with their environment, and this role that they play in conjunction with number 1, all 3, 1, 3, and 4 together help the cell to communicate with its outer environment and make changes inside. As I said in class, 2s aren't very common, and so for our purposes, we won't really talk about them. But 1, 3s, and 4s play very important roles in signaling. So signaling is something I leave at the very end of the course. So if you want to hear more, come by, I'll tell you more. Let's see, I had this question over here first. The inside is up, that's correct. Yep. Yes? So her question was, transcription proteins are proteins that control transcription, and she didn't have any examples that I expected to know examples. Well, I did talk about two examples. One was the lac repressor, right? That was a protein that affected transcription of the lac operon. I also talked about the cap protein, right? I talked about enhancers, right? Yeah, that's fine. Oh, sorry. Go ahead. Sequence elements are sequences in DNA bound by transcription factors. Huh? The transcription factors bind to them. And so they affect transcription. Okay, question? Yes? I didn't give a name to it. It's just called the -35 sequence. So it's another sequence that is common among all regions ahead of E. coli genes, okay? And it's recognized by the sigma factor of the RNA polymerase. Yeah. Yes? Say it again. I'm sorry. General structure of a nucleotide, yeah. I mean, a nucleotide has a sugar, a phosphate, and at least one phosphate and a base. Yeah. A simple sketch. Okay. I won't grade you on your artwork. Let's put it that way. Yes, back in the back. Of what, Sphingolipids? Yeah, sure. Sphingolipids are actually rather like, at least in their base structure, they're rather like the glycerophospholipids. Okay. This at the bottom shows you sphingomyelin, which is one of the sphingolipids. Okay. And sphingomyelin has a structure that looks like this, okay? Well, it comes from sphingosine, which has this structure right here. And no, you don't need to know the structure, all right? So you're not going to have to sketch a structure of a sphingolipid on the exam. But let me just, since you've asked the question, show you a little bit about its structure, okay? So I'm showing you some things that you won't need to know, but remember that a glycerophospholipid had a carbon, 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 and then fatty acid, and then a phosphate, right? All right? This guy has a carbon, 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 carbon. It's got a long chain, and it's got some kind of a chain here. Notice it has a phosphate down there also. That looks an awful lot like a glycerophospholipid at first glance. So structurally, they're rather related. All right? But you're not going to have to draw the structure, so don't worry about that. Now, her question had to do with function, and sphingolipids are molecules that are found in membranes, just like glycerophospholipids are found in membranes. And in particular, sphingolipids are found a lot in neural membranes. So remember we talked about cerebrosides and gangliosides? Those are sphingolipids that have either, in the case of a cerebroside, a very simple sugar like glucose attached at one end, or in the case of a ganglioside, has a complicated sugar attached at one end. And there's the end right there. Okay. Both of these play roles in neural tissue, commonly in the brain, for example. Okay? Yep. You're like the fourth person to ask me that question. Yes, a lot of interest in cholesterol, which is good. So cholesterol is needed in membranes. I haven't really said too much about why, but again, since you've asked me, I'll tell you, but you won't need to know. Okay? So remember that membranes, we want membranes to be fluid. But we don't want them to be too fluid, and we don't want them to be too rigid. Okay? So we've got a balance. That's why the cell adjusts, for example, in the case of fish, how much unsaturation it puts in to make them about the right balance for the temperature in which they exist. The other thing that cells can do to extend their fluidity is by putting more cholesterol into the membrane. Because cholesterol sort of extends the range of fluidity a bit.
So it can make them, the functions make them more.
Well, I just say it extends the range of fluidity. Instead of being a fairly narrow temperature range, it's a wider range. So I won't say it makes them more fluid because it doesn't change their fluidity, but it changes the temperature range over which they are fluid, makes it wider. That's what cholesterol does. You don't need to know that though. Okay? Yeah. I did talk about those in class, so you should know what that refers to, huh? Yeah. Okay, sure. So the transition temperature related to DNA and the melting of DNA. So when I talk about that, here. Okay. This is the graph that you're talking about, all right? Well, let's not look at the details of it. Let's just think about what's happening, okay? So what's happening? If I take a protein and I heat it, what happens to the protein? It denatures. So I can denature DNA with heat just like I can denature a protein with heat. Now, DNA, when I denature it, has a double helix. So if I denature it, I pull the strands apart. That's what I'm doing. All right? So if I add heat, I'm simply pulling the strands apart. In the case of the protein, I can't look in the solution and see anything different. I can't physically see something. But in the case of DNA, when I pull the strands apart, each strand alone absorbs more light than the two strands together. So if I take these two strands apart and I measure the absorbance, and I put the two strands back together and I measure the absorbance, when the two strands are apart, I'll have more absorbance than when the strands are together. Okay? Now, what's happening here is I'm increasing the temperature and the absorbance is going up. What's happening? The strands are coming apart. That's all it's showing you. Okay? So all that is just the midpoint, and that's what people talk about as the place where the strands are half apart. Yeah. So you talk about ice having a melting point. You can talk about DNA having a melting point. It's the transition temperature. Clear as mud, guys? One more question in the back.
Can you clarify the difference with that DNA polymerase 1 does and what DNA polymerase 3 does?
Okay? So the difference between what DNA polymerase 1 does and what DNA polymerase 3 does. All right? Let's look at the table that I gave you for that, and I will be happy to do that. Okay. Oh, that's eukaryotic. Sorry. I don't think you want to go through all that. Here we go. Okay. Polymerase 1, polymerase 2, polymerase 3. Polymerase 2, as I said, didn't talk about much in class, and I'm not responsible for it. I'll just tell you briefly here, polymerase 2 is involved in repair. Okay? Repair. Now, polymerase 1 and polymerase 3 I did talk about. Polymerase 3 has the primary function is it is the polymerase at the replication fork. It's making the leading strand, and it's making the lagging strand after the primase. It makes them in both cases after the primase because the leading strand is after the primase as well. Once primase does something, DNA polymerase 3 is going to extend it. So what is DNA polymerase 1 doing? DNA polymerase 1 has an activity that the DNA polymerase 3 does not have. The activity that the polymerase 1 has is it has a 5'. What happened there? I fixed it and I fixed it backwards. Oh. I hate it when I get in front of class and discover that's when the error is. These two are flipped. I don't know why that's flipped. Okay. In any event, what polymerase 1 has is a 5' to 3' exo. That 5' to 3' exo removes RNA. Polymerase 3 cannot do that. Polymerase 3 cannot do that. So what polymerase 1 does is it removes the RNA primers, replaces them with DNA, and then gets out of the way. So it's really important in Okazaki fragments because there's a whole bunch of Okazaki fragments, each of which have an RNA to start with. The RNA polymerase 1 is coming along those Okazaki fragments and fixing that RNA. Polymerase 3 can't do that. After DNA polymerase 1 has fixed that, DNA ligase comes in and ties everything together, and replication is complete. They both proofread. They both proofread. Polymerase 1, that's why this thing is backwards. They both proofread. They both have 3' to 5' exo. I wish I discovered that before now. Oh well. I'll fix it tonight. It'll be too late.
Did you mention that polymerase 1 has some sort of repair activity as well?
So polymerase 1 does participate in some repair processes, but they're just the ones I've showed you on the screen. 2 actually appears in other ones as well. Yeah. Other questions? All right, guys, let's see you raise that average. I want to see you get out there and nail it, okay?